This is Dr. Malti from Life Sciences Department. In this session, we are going to study about the mechanism of enzyme interaction and mechanism of enzyme action. So first we should know what is active site. So active site which will be present on the enzyme, which will be called as catalytic site, in which the substrate goes and binds, interacts and gets converted into product. So this particular site where the substrate goes and binds and gets converted into product, we call it as active site. Oxton in around 1948, he pointed out the interaction between enzyme and substrate. So the enzyme and substrate has three different points of interaction. So these three different points of interaction are very important for enzyme and substrate complex. To form ES complex, these points of interaction are very important and it is required. Only then two things are met. One is geometric complementarity with respect to shape. The other one is electronic complementarity with respect to charge. So in case of enzyme substrate interaction, we have 3 points of interaction between enzyme and substrate. So the substrate will have two binding groups, which is shown in the red color, and the enzyme also will have two binding sites. This is also shown in the red color. And in case of substrate, we have a reacting group, which is shown in yellow color, and in the enzyme we have catalytic group. So the binding group of substrate will be binding with the binding group of enzyme in such a way that the reacting group of substrate will be exactly communicating with the catalytic group of the enzyme, so as to form 3 points of interaction. So this 3 points of interaction, which include the reacting group, catalytic group binding, and two binding groups which will be interacting with the other two binding groups of enzyme. So for the enzyme substrate interaction, we have two hypotheses. One is lock and key hypothesis, which was proposed in 1890 by means of Fischer, and the other one is induced fit hypothesis in around 1958 by means of Koshland. So the first proposed hypothesis is lock and key hypothesis, and this is very simple hypothesis in which the enzyme and substrate interacts exactly complementary in its geometry. So the geometric complementarity of your enzyme and substrate, which will exactly resemble like a lock and key. So here the enzyme has the site, the catalytic site, which will be exactly complementary to that of the structure or the geometry of substrate, so as to have enzyme substrate interaction. So the direct interaction of enzyme and the substrate to form enzyme substrate complex. So later on, the complementarity of enzyme and substrate is not exactly complement when we see all the enzymes. So in order to fit that kind of interaction, we had Koshland's proposal in around 1958. In which the enzymes in free solution will not be exactly complementary to the substrate. When the substrate approaches the enzyme, the conformation of enzyme will change in such a way to fit the substrate molecule. The enzyme will be floppy and the substrate will be rigid, so that the enzyme can wrap around the substrate, so that the conformational change will be happening in the enzyme, whereas the substrate group will be as such rigid. So based on the structure of the substrate, the enzyme will fit itself into the substrate and it will form enzyme substrate complex. So during the formation of this enzyme substrate complex also, the reacting group and the catalytic group will be interacting with each other along with the binding groups. So in case of the induced fit model, we have the enzyme which is not complementary to that of the substrate. Now as the substrate is very rigid, the enzyme will flap around the substrate, especially the binding region, so that the complementarity will be attained by enzyme, since the substrate molecule is a rigid molecule, so as to get the enzyme substrate complex. So as the product leaves the enzyme will attain its original conformation as it was before the binding of the substrate. So next we have mechanism of catalysis of enzyme. So first we have acid-base catalysis of enzyme. So in case of acid-base catalysis, it will be the normal general acid-base catalysis of the particular chemical substance or the chemical reactions. So here what happens, as usual, the substrate will attain the transition state or intermediary state. From the transition state, the product will be formed. So in the transition state, the particular transition intermediates will be highly unstable. So in order to catalyze the particular reaction, the particular catalyst will go and stabilize the transition state intermediate so that the transition from the transition state intermediate into product will be faster. Usually this will be done with the help of water molecule which is present in the particular system. So the water molecule can act as either proton donor or proton acceptor to the particular transition state intermediate which is unstable. So in a case where the water molecule either donates the proton or withdraws the OH minus ion from the transition state intermediate and to this transition state intermediate, either one of the mechanism will be taking place so that the transition state intermediate will be stabilized so that the product formation will be faster when compared to the normal chemical reaction. If the water molecules are involved in such kind of intermediate stabilization, we call it as specific acid-base catalysis. In case if the protons or the OH minus groups are removed or added through the other molecules, especially through the weaker acid and weaker base, we call it as general acid-base catalysis. So the withdrawal or the donation of H plus to the transition state intermediate, if it is done through weaker acid or weaker base, we call it as general acid-base catalysis. So same acid-base catalysis will be done through enzymatic way also. So the enzyme's active site amino acid which can act as either acid donor or base H plus abstraction molecule. So any of the weaker acid and weaker base, whatever the function it is doing, the same function will be done by the R group of amino acids which are present in the catalytic site of enzyme. So same way, the proton donation or abstraction of H plus will stabilize the transition state intermediate so that the product formation will be faster. So usually in the presence of enzyme, the transition rate or the product formation rate will be enhanced around 10 to the power 2 to 10 to the power 5 times greater in the presence of enzyme as catalyst. So now in the reaction, in the general reaction, I had given the primary alcohol group and we can see OH and C double bond O. The alcohol group and the keto group are interacting. So during the intermediate formation, OH bond is not formed, it is in the transition state and this particular molecule is highly unstable. So in order to make this intermediate transition state to stabilize, either the OH will be abstracted with the help of water molecule so that hydronium ion will be formed, or with the help of base we can abstract the H plus ion. So once if we abstract the H plus ion, the other intermediate which is formed has COC bond. So now this particular intermediate compound can be converted into product very easily. So this can happen in the presence of water as well as in the presence of base. But in the presence of water, it is little more slower. So if we add base, especially the weaker base, the catalysis of the reaction will be faster when compared to the presence of water. So next we have covalent catalysis. From the name it's covalent catalysis. So covalent bond will be formed in the transition state intermediate. So example I had given generally AB, in which X will be acting as your enzyme molecule. So now what is happening is the intermediate which is formed AX has to be formed into product A plus B. The bond between A and B has to be removed or broken down. So in order to do so, the enzyme will be acting as a nucleophilic group. So nucleophilic group which will have negative charge and it will go and attack the positively charged species. So now the enzyme will be acting as nucleophile. It will go and interact or attack the substrate groups so that the covalent bond will be formed between the intermediate and the enzyme. So now in order to have the product formation easier, this interaction of covalent bond will lower the activation energy than the uncatalyzed reaction. So the new step will bring a faster product formation when compared to uncatalyzed reaction. And here the nucleophile only will be formed from enzyme cofactor or from the amino acid side chains. So these only will be interacting and forming the covalent bond with the substrate molecule. So once the covalent complexes undergo further product formation, automatically the free enzyme will be released, and the free enzyme along with the product which are formed. So the last mechanism of action is through metal ion catalysis. So this also will be almost similar to covalent catalysis, but here the interaction will be non-covalent interaction. And here the enzyme bound metal ions which are present in the enzyme will participate in catalysis process. And here the substrate will be interacting, or the substrate will be stabilized with the help of your enzyme's metal ion. So the interaction will happen between the metal ion and the substrate's transition state. And here now the weaker interaction will be happening. So weaker ionic interaction will be there between the metal which is present in the enzyme and the substrate molecule. So here also the reaction will be similar to enzyme substrate binding, but the substrate transition state will be interacting. Metals also can mediate oxidation and reduction reactions which are reversible in nature. In case of this metal ion catalyst also, once the product is formed, the enzyme's original conformation will be attained. So using covalent catalysis, general acid-base catalysis, transition state stabilization, the chymotrypsin mechanism will be governed. Thank you.